Well, lipoprotein little a is a genetic risk factor, and until recently, there hasn't been a way to treat this, partly because small molecules and antibodies don't work very well to lower the levels. And so it has to be a convergence of some new therapies that come along that allow novel therapeutic ways to treat diseases. So lipoprotein little a is one of these. And it aims to treat a very large segment of the population ultimately, because about 20% of the population as a whole has elevated levels. So if you extrapolate this to globally, it's about 1.4 billion people. And a lot of them don't know they have it because most doctors don't check levels. Now, not all of those patients are going to get cardiovascular disease, but a large proportion of them do. So this addresses the need where you have your LDL controlled, your triglycerides controlled, everything else is good, but you still have this additional atherothrombotic risk factor that hasn't been addressed yet. So up to now, we have no therapies for it that are approved, and this approach addresses that significant unmet need in a large segment of the population. So it's an antisense oligonucleotide, and it's in the realm of RNA therapeutics. And right now, there are about 6 drugs only approved as RNA therapeutics. So this is a very young field, and because of that, there's not a lot known about it. But the bottom line is that we all know through classical genetics that DNA makes RNA, which makes protein. And most therapies are targeted towards proteins to inhibit their function or get rid of them. What this does is actually inhibits the mRNA from making protein. So it works at the factory that makes something you don't want to make too much of. So the drug is injected subcutaneously. It goes to the liver in this particular case. It gets into the cell and binds any mRNA that makes lipoprotein little a. And when it binds it, there's a natural process in the cell that cleaves the mRNA so it can't make protein. So you're basically shutting down protein production. And to give you an example of other approaches, PCSK9 antibodies bind the protein that's already made. This goes a step earlier, prevents the cells from making the protein in the first place. So it's a very elegant way to treat diseases of protein overproduction. So we're presenting a phase 2 trial in patients that have cardiovascular disease, so heart attack, stroke, peripheral arterial disease. We have also twice normal elevated LPA levels at least. And so we did 5 different doses. We had a placebo group. They got subcutaneous injections, either monthly, every 2 weeks, or once a week. And then we followed from 6 to 12 months. And that's basically the trial design. That should reflect the kind of patients that ultimately we want to treat with this drug. And what we showed is that the drug works very well. It lowers Lp(a) up to 80% in the highest dose. And this is unprecedented because up to this point, we've had niacin and PCSK9 inhibitors that lowered about 15 to 30%. So this is a home run in terms of the efficacy of the drug. We also looked at some additional measures. One was, how many patients can you get to a level below a certain threshold that's felt to be safe? And that threshold is under 50 milligrams per deciliter. And at the highest dose, we had 98% of patients that achieved a threshold where the Lp(a) has very little risk. And this is also a very important metric because what you can say is everybody will respond and get really good levels. You don't have to give like multiple different doses to get where you want, or multiple drugs. This one drug can pretty much take care of the majority of patients that have elevated Lp(a). We did some additional measurements. We measured oxidized phospholipids with an assay that we developed at UCSD where I work. That assay reflects the pro-inflammatory effects of Lp(a), and those were down between 60 and 80%. And the drug actually also lowered LDL cholesterol about 21% and ApoB100 about 15%. And both of those particles are atherogenic. And so if you look at all that data, it looks really promising. You have LPA reduction, oxidized phospholipid reduction, LDL reduction. All those are bad guys. They're all going in the right direction. We looked at a variety of safety endpoints and noted there were no issues in platelet counts, in renal function, no significant issues in liver function. The drug looked very safe. There was only one dropout from injection site reactions. And there didn't seem to be an imbalance in adverse events between the treated groups and the placebo groups. So, it's 286 patients, 6 to 12 months of follow-up. It's not a giant study, but nonetheless, it's suggestive that it should be fairly well tolerated as we go forward to the next part of the development. Well, we can have several conclusions. One is that we finally have a therapy for what was thought to be an undruggable target, which is Lp(a). So this is very important. It's sort of like the equivalent of having the first statin, except instead of this being a relatively weak drug like the first statins were, this is about the most potent drug you can make for this because you can get 98% of patients to goal. So that's one lesson number one, a very effective drug. The other lesson is that it also affects oxidized phospholipids and LDL, so there could be like a 3-for-1 benefit here because it does have additional effects beyond lowering Lp(a). So this will allow us to go to the next step because since this drug works so well in phase 2, now the next step is to do a phase 3 outcomes trial where not only do you look at these biomarkers, Lpa, oxidized phospholipids, LDL, you're going to look at cardiovascular events. And the question now is, if you lower Lp this much and you get almost everybody to what you think is a normal level, will there now be less heart attack, strokes, and other cardiovascular events that are translated to clinical benefit?